excited for the microscopy. Let's move this one and this one. So, first, we are going to observe a living daisy. Now, oh, hello. We have a little friend. A weevil. A weevil. Here, look. I found a cutie. So cute. I think I scared him. He's not moving. So cute! A weevil! It's a weevil! I think it's a beetle from the coleoptera. I'm not sure. Do you see how the light is moving? That is water, and the fact that the water is capable of staying on that much is for two main reasons. First, all the characteristics of water. Second, these little hairs on the roots. They have two functions, but the first one, absolutely important and fundamental, is to magnify the surface of absorption. I present you the root tip. This is specifically from a plant called the mice. The mice. The one for the popcorn. So, these are all the cells. Why it's not symmetrical? Shoot! Now, you see how much are they small? Oh, guys! The colors you see, they are not the one from the actual plants. They are the tint you use in the lab in order to make the sample more visible or clearer. Did you remember we were talking about the stomata? On the lower part of the leaf, they are made of two major cells and two guardian cells. They open and close based on the water pressure inside them. So, if the plant is all okay, has a lot of water, okay, she can open the stomata in order to breathe and in order to start a lot of cute reactions. So cool! Oh my god! It's like a disco party for bacteria! From bacteria! Why are they organized as puzzle pieces if hexagons exist? Oh, because with hexagon, you are going to have a very straightforward perimeter for each set, while with these other methods, so like puzzle pieces, you are going to have the same volume, maybe, but a way bigger surface. And if you have a big surface, that means you can have more and more point of contact between a cell and another. They are way more locked together and they can exchange a lot more of information and molecules. Now, this is the peel of an orange. Can you guess what are these little holes we see in the glass? When you squeeze an orange peel, the droplets that come out are still there. Yes! Oh yes, they burn super nicely. They are a very big deterrent for cats. This is a pine leaf. So, wait, I think I found it. Let's see if I can zoom even more. Yes!
It is so beautiful, it feels fake.
It's not.
I know. It looks like a 3D image.
Is that sila? Oh no, that was a mushroom. That thing? Oh, a mushroom. So, sounds like there needs to be another microscope stream. Oh! You enjoyed that much? Oh, shoot! Mushroom! I am really happy you like that. So, let me smooch you all. Mwah! Have a super beautiful evening and have a lot of fun. Mwah! Mwah! A super goodbye you all.